Hey everyone, and welcome to another deep dive.
Yeah.
Today we're going to be talking about spectroscopy.
Spectroscopy.
And I think this is going to be a really cool one because I think it's something that a lot of people I've heard of, but maybe don't really
Yeah.
understand. And it's used so much in
It's everywhere.
so many different
You
fields.
know, it's really everywhere. And I think in the pharmaceutical industry, it's probably one of the most common techniques used.
So let's just dive right in.
Yeah, let's get into it.
So basically, spectroscopy is
Using light to analyze matter.
okay. It's like if you shine a light through a prism,
Okay.
right? You get a rainbow.
Yep.
And all those different colors are different wavelengths.
Different wavelengths, exactly.
And so it's kind of like that, but instead of just looking at the colors, you're analyzing like how that light is interacting with whatever substance you're looking at.
Yeah, you got it. So every compound interacts with light in a very specific way.
Okay.
And by analyzing that interaction, we
Uh.
can learn a lot about the substance.
So what are some of the ways that,
Yeah.
what are some of the types of spectroscopy, I guess?
Yeah, so there are a lot of different types, but some of the main ones that we're going to talk about today are UV-vis spectroscopy or UV visible spectroscopy,
Okay.
infrared spectroscopy,
Okay.
nuclear magnetic resonance spectroscopy, which we call NMR,
NMR, got it.
and then mass spectrometry, which is MS.
Okay, so we got a lot to cover here.
Yeah.
So let's start with like UV-vis spectroscopy.
Okay, so UV-vis spectroscopy.
What is that all about?
It's great for determining the concentration of drugs and identifying compounds with specific structures.
Okay, so like if I have like a solution and I shine UV-vis light through it,
Yeah.
I can tell how much of a certain substance is in that solution.
Exactly.
Yeah.
Yeah. And you can also look at like if a compound has double bonds or triple bonds.
Oh, interesting.
That sort of thing.
Okay, so it's kind of like I don't know why I'm stuck on analogies today,
No analogies are good.
but it's kind of like a fingerprint almost.
It is like a fingerprint. Yeah.
Like every compound has a
has a unique
way that it interacts with
with the light.
that UV-vis light.
Yeah.
Okay.
And that's how we identify it.
Okay, cool. So that's UV-vis. What about infrared spectroscopy?
Okay, so infrared spectroscopy or IR spec is kind of like molecular fingerprinting.
Molecular fingerprinting. Okay.
So every compound has a unique pattern of infrared light absorption that reveals its functional groups.
Now, what's a functional group?
So a functional group is basically just a specific group of atoms within a molecule
Okay.
that determines how the molecule behaves. So for example, an alcohol group, which is OH, will absorb infrared light differently than a carbonyl group, which is C double bond O.
Okay. So it's kind of like looking even deeper into the molecule
Exactly.
than just...
It's more specific.
Yeah, okay, cool.
Yeah.
All right, so IR spectroscopy is like the fingerprint expert, I guess?
Yeah, you got it.
All right, what about NMR?
Okay, so nuclear magnetic resonance spectroscopy.
That's a mouthful.
NMR is like getting an X-ray of a molecule.
Whoa, okay.
So you can actually see the detailed structure, particularly the carbon and hydrogen framework.
Oh, wow. Okay,
Yeah.
so that's like really detailed.
Very detailed, yeah.
All right, so we've got UVVs, we've got IR, we've got NMR.
Uh-huh.
What about mass spec?
Okay, so mass spectrometry, or MS,
Mm-hmm.
it's a little bit different.
Okay.
Imagine a molecular weighing scale.
Molecular weighing scale.
Yeah.
Okay.
So it separates ions based on their mass-to-charge ratio.
Mass to charge ratio. Okay, I need you to break that down for me.
Okay, so every atom has a specific mass, right? And when an atom becomes an ion, it either gains or loses electrons,
Okay.
which changes its charge.
Okay.
So the mass-to-charge ratio is basically just the mass of the ion divided by its charge.
Okay.
And mass spectrometry can differentiate between molecules based on even tiny differences in their mass-to-charge ratios.
Okay, so we're talking about like really, really tiny differences here.
Yeah, it's very precise.
Okay, so we've got like these four main types of spectroscopy.
Yeah,
But you mentioned before that like each compound kind of has its own fingerprint.
right.
What does that look like?
So we call these fingerprints absorption and emission spectra.
Okay.
So imagine you're shining a light beam through a colored solution.
Right.
Some wavelengths of light will be absorbed by the solution,
Okay.
while others will pass through.
Okay.
So the color that we see is the result of the wavelengths that weren't absorbed.
Okay.
So the pattern of wavelengths that are absorbed
Uh-huh.
forms what we call the absorption spectrum.
Okay.
And that is unique to that particular compound.
Okay.
So it's like the compound signature
Oh, okay. I get
on
it.
the light beam.
So what about the emission spectrum?
Okay, so the emission spectrum is when a substance is energized,
Okay.
maybe by heat or light.
Okay.
It can release some of that energy as light,
Okay.
and the pattern of wavelengths that are emitted
Yeah.
forms the emission spectrum.
Got it.
And that is another unique identifier for the compound.
Okay, so we have the absorption spectrum, which shows like what colors are absorbed,
Yes,
and then the emission spectrum, which shows what colors are emitted.
you got it.
And both of those are like fingerprints for different compounds.
Exactly.
Okay, I think I'm starting to get this. So like, how are these fingerprints actually used,
Yeah.
like in the real world?
Okay, so let's go back to our aspirin example.
Okay, back to aspirin.
So remember how we talked about making sure that a batch of aspirin is pure?
Right.
Well, we can use spectroscopy to do that.
Okay.
So by analyzing the absorption or emission spectrum of the aspirin sample, we can confirm its identity and check if there are any impurities present.
So it's like matching fingerprints, basically.
exactly.
If the fingerprint of the sample matches the fingerprint of pure aspirin, then we know we're good.
Then you're good to go.
Okay, cool. And this is used for way more than just aspirin, right?
Oh, yeah. Let's talk about how spectroscopy is used in a more modern setting, like pharmaceuticals.
Okay, yeah. So back to medicine.
Back to medicine.
So how does spectroscopy help with like drug development and quality control?
So let's say you're developing a new drug.
Okay.
You've synthesized a promising molecule.
Yeah.
But you need to make sure that it's the right structure
Okay.
and that it's pure. So we use techniques like NMR and mass spectrometry.
Right, because NMR gives you like that 3D structure.
Exactly.
Aspect tells you like the mass and if there are any impurities.
Yep, you got it.
Okay.
And then once the drug is approved, we use spectroscopy for quality control.
Oh, right, right. Because you got to make sure that like every batch
Every batch is consistent.
is the same.
Yep.
And it's like safe and effective.
Exactly.
Right.
So it's basically like a molecular inspection.
A molecular inspection.
Making sure that each pill or each vial
Yeah.
has the right amount of the active ingredient
Right.
and that there's no contaminants.
Okay.
So it's used for both qualitative and quantitative analysis.
Okay, cool.
So yeah, there are so many applications of spectroscopy.
Yeah, it seems like it's used for pretty much everything.
Pretty much. Yeah, it's a really versatile tool.
So we've talked about how spectroscopy helps us analyze and identify things.
Right.
But can it also tell us like how much of something there is?
Yes, absolutely.
Right.
So remember how we talked about UV vis spectroscopy being used to measure the concentration of a drug?
Uh-huh.
Well, the amount of light that a substance absorbs
Okay.
is directly proportional to its concentration.
Okay.
And this relationship is known as Beer's law.
Beer's law.
Okay, it's a very important law in spectroscopy.
I'm guessing it's named after someone who likes beer.
Maybe.
So basically, the more of something there is, the more light it absorbs.
Exactly.
Okay, that makes sense.
And by measuring the absorbance at specific wavelengths
Okay.
and applying Beer's law,
Uh-huh.
we can figure out the concentration of the substance.
So it's like a molecular measuring cup.
You could say that, yeah.
That's pretty cool.
It's pretty cool.
So we've got these amazing tools for qualitative and quantitative analysis.
Yeah.
What's next for spectroscopy?
What's the future hold?
Yeah, like what are some of the most exciting developments that are happening?
Well, one area that's really taking off is combining spectroscopy with imaging techniques.
Imaging techniques.
Yeah,
So like instead of just looking at a single point,
right.
you're getting like a whole picture.
Exactly. So you can visualize the distribution of different compounds within a sample,
Oh, wow.
which can give you really valuable insights into complex systems.
Okay.
So for example, in medicine,
Yeah.
this could be used to track how a drug is delivered to specific tissues
Oh, wow.
or to monitor the progression of a disease.
So it's like taking spectroscopy to a whole new level.
Exactly.
That's awesome.
Yeah.
Are there any other advancements that you're particularly excited about?
Yeah, another one that's really cool is the development of portable and miniaturized spectrometers.
Hey, so you're saying we can take these things out of the lab?
Yeah, out of the lab and into the field.
That's all.
So you could use a handheld spectrometer to analyze the soil in a field
Oh, wow.
or to test the water quality in a disaster zone.
So it's like giving anyone the power of spectroscopy.
Pretty much.
That's incredible.
It's pretty exciting stuff.
Yeah, so spectroscopy is all about unlocking secrets.
Unlocking secrets.
Secrets of the past, secrets of molecules.
Secrets of the world around us.
Exactly. And the best part is we're just getting started.
This has been such a cool deep dive.
Yeah, it's been fun.
So before we wrap things up, let's just recap the key takeaways from today.
Okay, good idea.
Okay, so to recap, we've learned that spectroscopy is like using light to analyze Like anything, really.
Yeah, it's like a molecular flashlight,
Molecular flashlight. Yeah, that's a good way to put it.
shining a light on the molecular world.
Exactly. And we talked about all the different types of spectroscopy.
Right, UV, infrared, NMR, mass spectrometry. Well, this deep dive has been incredible.
Yeah, it's been a lot of fun.
We've learned so much about spectroscopy,
We have.
from ancient pottery to the stars.
Yeah, it's a truly fascinating field.
And it seems like it's just going to keep getting more and more exciting.
Definitely. There's so much more to discover.
Well, I'm already looking forward to our next deep dive.
Me too.
But for now, let's leave our listeners with something to think about.
Okay, so the next time you see a rainbow,
Okay.
remember that it's an example of light being separated into its different wavelengths.
Light, like with a prism.
Exactly.